VWD is a genetic clotting disorder in dogs, where dogs have a deficiency of the blood glycoprotein von Willebrand factor, VWF, which is crucial for hemostasis. This glycoprotein has two main functions: to mediate the adhesion of platelets and carry factor VIII in plasma. Platelets are small fragments that range from 2 to 4 micrometers in size and assist with clotting blood at sites of vascular damage. The VWF genes bind to specific membrane glycoproteins on the platelets and to subendothelial proteins in endothelium or inner layer of blood vessels. These form the crucial blood clots to prevent excessive bleeding from a vessel. VWFs also stabilize and carry factor VIII in plasma around the body, which is important for fibrin clot formation. A lack of the normal VWF gene has a great impact on dogs, meaning if the dog becomes injured, there is an inability of the body to naturally reduce blood loss, as injured vessels are not healed. There are 3 types of von Willebrand's disease. Type 1 is the most common, occurring in 70 to 80% of affected animals, and it is a quantitative defect, meaning there is a reduced production of von Willebrand factors. Type 2 occurs in approximately 20% of individuals and is far more severe, being qualitative, meaning there is a functional issue of the VWFs. Type 3, found in less than 5% of cases that have this disease, is quantitative and is distinguished by the dog having a complete absence of VWFs. Von Willebrand's disease is a heterogeneous autosomal genetic disease, which means it is accounted for by any one of a multiple number of alleles or non-allele locus mutations, and it is passed on through families but not by the sex chromosomes. Hence, it is a genetic disease. The mortality of this disease is low, however the morbidity is high, and it is transmitted as an incomplete dominant trait, which is why the severity of this disease can vary in 3 different types. It is most commonly found that types 1 and 2 have an autosomal dominant pattern, whereas the more severe type 3 has an autosomal recessive pattern, which explains why it is rare in dogs with VWD. The VWF gene is found on the short arm of the 12th chromosome, and its gene has a length of 178 kilobase pairs. The resulting mRNA strand copied from this gene is expressed by endothelial cells, the inner lining of blood vessels, and bone marrow megakaryocytes, the cell type in bone marrow that produce platelets. In types 1 and 3 of VWD, there are major disruptions within the gene that accounts for the von Willebrand factors, including deletions, where part of the DNA is lost during replication, nonsense mutations, where the DNA is changed to a chain-terminating codon, and point insertions, where one base is deleted and another is inserted in its place. These disruptions greatly affect the synthesis of the VWF gene. These newly created VWF genes are called null alleles, which are alleles that are a mutant copy of a gene that completely lacks its normal function. These null alleles are present in a homozygous state, for example, X, or lowercase x, resulting in type 1 von Willebrand's disease, or a heterozygous state, X, x, resulting in type 3 of the disease. Many carriers of type 1 disease with lowercase x, are found to be normal in every aspect, but may pass the disease on to their progeny if paired with another carrier. Type 2 is similar, however the function is affected due to the lack of intermediate and large forms of von Willebrand factors in the plasma. Moreover, there are 3 spontaneous mutations that prevent the assembly of the propeptide that catalyzes the formation of large VWFs. These mutations present a recessive phenotype that affects the function of the VWFs. Therefore, type 2 will present itself with every case as opposed to type 1, in which the dog may appear normal but still carry the disease.
The aforementioned leads into an epic consideration in ethics based around the appropriate breeding of affected parents or carrier parents. Major breeders and dog farmers financially lack the incentive to test for the presence of von Willebrand's disease. As a result, the detrimental phenotype is often expressed in future generations. Due to this, the only way to ensure the continued breeding of dogs without this debilitating disease is to test all the parental generations and progeny that is descended from them, and cease breeding from any that either display the phenotype or carry the genotype. This act is recommended from a veterinary perspective as it focuses on the best interest of the animals. All parentals showing the VWF phenotype or carrying the genotype should no longer be used for breeding, which will ensure loss at the cost of the breeder. In an attempt to establish the best compromise between welfare expectations and breeders' financial stresses, a government or private funding is basically the only option. As for the owners of animals whom have von Willebrand's disease, the main ethical consideration is the quality of life of the animal and ensuring it lives a pain-free and normal life. The concept of euthanasia for victims of von Willebrand's is generally not considered, as most animals suffering from this disease can still live a happy life. Von Willebrand's disease is treated through several ways, including desmopressin, recombinant von Willebrand factor (RVWF), von Willebrand factor, antifibrinolytic drugs, and platelet transfusions. Research into gene therapy is currently the most promising avenue to attain a potential permanent cure of this genetic disease. Despite this promising outlook, the lack of prevalence of von Willebrand's disease and its clinical signs leaves it lacking in priority ranking for gene therapy trialing. Furthermore, point mutation has been attempted, but with little success. Other research on this heterozygous autosomal disease is yet to be carried out, but with increased breeding of dogs, including inbreeding and other genetically detrimental practices, genetic diseases such as this are becoming more prevalent. Ultimately, as time goes on, veterinarians will be made to more frequently treat genetic disorders such as von Willebrand's disease. This suggests that a more aggressive and active approach to researching and finding permanent genetic therapies should be at the forefront of the collective mind of the community.